So this is the process of glycolysis. Again, the splitting of glucose into two molecules of pyruvate, which occurs through these biochemical processes. Now, after we have the generation of pyruvate, those pyruvate molecules have to enter something called the Krebs cycle. The Krebs cycle is also known as the citric acid cycle or the TCA cycle, which stands for acetyl coenzyme A cycle. The pathway has 8 separate reactions, and each of those reactions are also dictated by a specific enzyme, just like we saw with the glycolysis pathway. However, in the Krebs cycle, we only generate 2 molecules of ATP, or 2 molecules of adenosine triphosphate. The importance and significance of the Krebs cycle is to generate what we call electron carrier molecules. Those electron carrier molecules are referred to as NADH, H+, and FADH2. These products are formed in the Krebs cycle and are used in the next cycle, which is known as the electron transport chain. Now, like I said before, I don't expect you to know all of the various conversions of molecules that exist in this pathway, but you do need to know that NADH, H+, FADH2 are generated through this process, and that's the key reason that this the Krebs cycle is included in the process of cell respiration. Now, the process itself will take place in the mitochondria, and once the Krebs cycle has finished, the electron transport chain is the next biochemical pathway that will take place in the process of cell respiration. If we are talking about prokaryotic organisms, and prokaryotic organisms are going to be things like bacterial cells, then the Krebs cycle is going to occur in a different location. It will occur in the inner surface of the cell membrane of a bacterial cell. So here's a diagram outlining what happens in the Krebs cycle. And I know this looks really scary because there's a lot of structures in here, but really when you think about it, it just is simply a series of conversions that take place. So we start off with our 3-carbon molecule of pyruvic acid. And what happens is that pyruvic acid is going to lose a carbon. And when the carbon is lost, you have the loss of a hydrogen that's going to be taken up by a molecule known as NAD+. So NAD+ is going to pick up a hydrogen that's been lost when pyruvic acid loses a carbon molecule. And this carbon molecule is going to bind with oxygen and generate CO2. So CO2 is going to be released into the cell as a waste product, or in this instance inside the mitochondria as a waste product. So once this pyruvic acid has lost its carbon, that allows the opportunity for coenzyme A to affix itself to the 2-carbon chain. So we have acetyl coenzyme A, which comes in and affixes itself to it, and then the acetyl coenzyme A actually gets converted, and so you have coenzyme A that is going to affix itself to the carbon. In the next step of this process, you have the formation of citric acid, which takes place when the acetyl coenzyme A combines with a form that's known as oxaloacetate. And that's going to be generated typically at the end of the cycle. But once those end products are fused with the citric acid, with the acetyl coenzyme A, bound to these 2 carbon structures, then you have the formation of citric acid. And then again you have the loss of a carbon, and that carbon will bind to carbon dioxide or to oxygen, so that way it can formulate carbon dioxide. And then you also have the uptake of another hydrogen taken from the citric acid molecule. That takes this 6-carbon sugar here and conforms it into a 5-carbon compound. And again, the process continues. So you release another carbon through the assistance of enzymes. releasing carbon dioxide into the system, and then a hydrogen is taken up by NAD+, being removed from this 5-carbon compound. And so now adenosine phosphate comes in, and it's going to pick up a phosphate group from the compound that's generated, and that's going to release a product ATP. So again, remember, only one ATP molecule, or only two ATP molecules are going to be generated in this process, because the process happens twice, but only one is generated when you use one pyruvic acid. So just remember that this process happens in duplicates. So one ATP molecule is generated, and that 5-carbon compound is converted from 5 carbons into 4 carbons through the loss of a phosphate group, the loss of a hydrogen group, and then the loss of a carbon group, all at this process. And then finally, another carrier molecule, FAD, comes by the 4-carbon compound, and it rips off 2 hydrogen molecules, so that way it can generate its own carrier molecule. So the 4-carbon compound loses 2 hydrogens, and then it will also lose an additional hydrogen to NAD+. So in total, we've got 3 hydrogens that are being lost, and that's going to generate a structure known as oxaloacetate, which will be used to generate citric acid. So again, don't get bogged down in the details of what happens in this particular cycle, but just know that the outcomes of the Krebs cycle are the fact that you start off with a pyruvic acid and you are generating electron carrier molecules by essentially removing hydrogens from these carbon molecules. So anything that's highlighted, NADH, NADH, FADH2, NADH, NADH, those are the end products that we want to get out of the Krebs cycle. We also get we only get 1 molecule of ATP and if this Krebs cycle undergoes 1 process. Remember, we're actually getting 2 molecules because the Krebs cycle has 2 pyruvic acids that it needs to use to generate these electron carrier transport molecules. Now, once these electron carrier molecules, NADH and FADH2, are generated, they're going to move on into the electron transport chain. And in the electron transport chain, here we have a series of oxidation reduction reactions where energy is released as electrons as they're transferred from one compound to another. There's a lot of enzymes that assist in the passing of these electrons. Cytochrome oxidase is an enzyme that's used to make sure that electrons are being transferred to oxygen. And oxygen is where the final electrons is the final acceptor for electrons. And through the process of the electron transport chain, we're able to generate a lot of ATP molecules. So with electron transport chain, we're typically looking at the generation of 32 to 34 inside of a cell such as ours that has a membrane-bound nucleus, or eukaryotic cell. And bacterial cells, which are prokaryotic, they'll generate about 34 products of adenosine triphosphate, which is the ultimate goal of this particular chain. So to give you guys a kind of a break of listening to me talk about this, let's listen to a pretty nifty song on it. So I hope you guys enjoyed that, because I know I did, and I definitely probably would have given these girls an A in the class for their creativity. So anyway, so now we're going to hopefully move into discussing more of the electron transport chain in a little bit more detail. But like I said, I'm more interested in you guys knowing the overall processes and kind of what's generated as end products for these particular mechanisms. So again, a large number of ATP molecules are going to be produced by the process of oxidative phosphorylation, which is basically adding oxygen and adding phosphates to specific molecules. It's a very efficient process because you're generating so much ATP through this process. And what those girls were singing about were basically all of these components: complex 1, complex 2, complex 3, complex 4 ATP synthase. Okay, so you have, again, these electron transport molecules. NADH is bringing in a hydrogen that's going to be used to open up this complex 1, and once that complex 1 is opened up, you have the generation of NAD plus molecules, but you have passings of the hydrogen ion through the membrane, which is going to signal the generation of the start of the electron transport chain. And so complex 2 basically gets stimulated by FADH2, and as that's stimulated, it's going to activate complex 3, where you're going to have hydrogen also passing through the complex. Once